Moving forth with bacteria. First of all, I'd like to mention that we have two types of cells. If we take cells as a broad spectrum, we have two types of cells. The first type is known as a eukaryotic cell. A eukaryotic cell, and I'm pretty sure you've guessed the other type. We have a prokaryotic cell. Eukaryotic cells and then prokaryotic cells. Eukaryotic cells have a nucleus and membrane bound organelles. Have a nucleus and membrane bound organelles. Prokaryotic cells have no nucleus and no membrane bound organelles. That's the basic idea. Now I'm going to list all the structures that you're going to find in a bacterial cell. Now please do note, you don't have to go into too much detail. Now, like we did for plant and animal cells, you don't have to know about each specific organelle at all. All you have to have is a basic overall idea of all the structures that can be present within a bacterial cell. So let's get to drawing. It's going to be a nice diagrammatic representation. I'm going to start off with a rectangle. Also do note, it's not going to be a strict rectangle at all. So I doubt it would turn into a rectangle. Look at that, turn into a square. My God. Hold on. And space it on top. And enlarge things a little bit. Yeah. Okay, I know it looks very boxy. It's not very boxy. It has a little curved end to it and curved end to it, and so right. Starting off, I'm going to divide this in half. Down this way, I'm going to divide it like this. You'll see why later. On the left hand side, I'm gonna give this bacteria something like this. That didn't come out that great. Let's do it like this. Okay, that's better. Write this. Something like this. Now I'll go into detail of what that is. And on the same side, I'm going to give it something like this. That structure you might be able to identify all of a sudden. I think I'll label using this color. Yeah, that works. That is a flagellum. That is a flagellum. And what are these things? This is just like a plant cell, the cell wall. The important thing about the bacterial cell wall is it's not made out of cellulose. Only plant cell walls are made out of cellulose. The bacterial cell wall is made out of something known as peptidoglycan. This is sometimes also referred to as murine. Do keep that in mind. That is the cell wall. And amidst the cell wall, what we always have is the cellular membrane. Cell membrane, cell surface membrane, plasma membrane, cytoplasmic membrane, anything. And before I forget, I'm going to label the cytoplasm. That is something I always forget. So don't make the same mistake. We've got the cytoplasm like so. Okay, now what did I draw? What is this excess structure? This excess structure is something known as a capsule. Something known as a capsule. It's basically like a coat, almost a protein coat. Now I divided this cell into two. No, why did I divide it into two? On the right hand side, you will find the always present structures. You'll find the always present structures. On the left hand side, I have for you the additional structures it can have. So for example, do all bacteriums possess a cell membrane? Yes, they do. Do all bacteriums or bacteria, my plurals are getting out of hand, bacteria have a capsule? Do all bacteria have a capsule? No, they don't. Some might not. But they surely, all of them have a peptidoglycan cell wall, a cell membrane, a cytoplasm, but not all of them have a capsule or a flagellum by any chance. An additional structure to all of these things would be, now these are prokaryotic cells. No, bacteria, they don't have a nucleus, so please do not draw a nucleus inside the bacterium, okay? Then where exactly are they storing their genetic material or DNA? It's just outside, like this. We call it naked DNA, which is circular DNA. Naked circular DNA. And the bacterium contains ribosomes. No big deal. Protein synthesis is required, so they have ribosomes. And apart from naked DNA, they have circular DNA as well. They will always have naked DNA, that's why it's on the right hand side. They will always have ribosomes, that's why it's on the right hand side, it's labeled on the right hand side. Apart from the naked DNA, they can have another form of DNA that looks something like this. We call that like this. It's not circular. These are plasmids. They are also circular DNA, yes. But the thing with these circular DNA is that they are in a small ring form known as a plasmid, and you can, here's the fun thing and cool thing about plasmids, you can easily extract plasmids. You can take this out and do whatever you want, and that is the basis of genetic engineering and agricultural improvements. These plasmids can do a lot. Bacteria are typically used as vectors. There's something known as a vector. You will learn about these things in a little bit more detail as you progress through this course, but all you have to know is these plasmids can be easily extracted from a bacterium, placed into some other form of organism, and thus we have the ability to alter genetic material. It's a very interesting field if you are interested. Molecular biology, as always, and plasmids are a key root of it. Apart from flagellum, they can have something else for attachment and movement and stuff. Those things are called pili. Pili or pili, or sometimes it's double L, sometimes it's just a single L. Pili is a controversial structure, not a problem. All you have to know is we have these things known as pili. They're almost like flagellum, but they're pili. And I'll talk about an infolding. For that to happen, I'm going to have to delete or erase this place and redraw in something like this. So this inner cellular membrane will not always be perfect. Sometimes it can feed into the bacterium like this, like so. We typically call, when that occurs, we typically call that an infolding of the membrane. We call that the infolding of the membrane. Why are these infoldings important? Sometimes within these infolding membranes, photosynthesis can happen. Sometimes photosynthesis can happen, or there's this process known as nitrogen fixation that can occur. There are a special group of bacteria known as nitrogen fixing bacteria. In their infoldings, mostly what happens is nitrogen fixation. Not photosynthesis, but nitrogen fixation. On the other end of that spectrum, we have more bacteria who are going to be better at photosynthesis than nitrogen fixation because they have certain other enzymes within these enfoldings that promote more photosynthesis than this nitrogen fixation thing. All in all you have to know is within these enfoldings, interesting things can occur. That is about bacteria. We'll talk about viruses. There's basically nothing to talk about viruses. It's very simple. Viruses are considered as being non-cellular or acellular. They are non-cellular or acellular parasitic organisms. They are non-cellular or acellular parasitic organisms whose only living feature is reproduction. All in all, the only thing that is going inside a virus's head is reproduction. Funny, right? All it's trying to do is replicate itself, duplicate itself, and just spread all throughout every organism, basically. I can't really give you a precise size when it comes to a virus because viruses vary in so much size, but I'll give you a general idea. Generally, you can find a virus ranging from 20 all the way up until 300 nanometers in size. 20 to 300 nanometers in size. Like so. I'll draw the rough structure of a virus. 2, 3, 4, 5. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. That's not a precise value, I just counted. Genetic material, and we have these little spike proteins type of things attached. Like that. This is what is referred to as being a capsid, and then this is the genetic material of that. Here's the deal. When it comes to genetic material of viruses, it can either be DNA or it can be RNA. It's not both. It's never both. Okay, DNA or RNA. Keep that clear distinction in mind. They can have DNA, they can have RNA, but it's going to be one of those things. It's never always both of them. Half DNA, half RNA. No, that's not possible. That's not going to happen. And there's something known as the Baltimore classification of viruses. Let me write that term down. Baltimore classification of viruses, and that classification utilizes whether the virus has DNA or RNA, whether the DNA is single-stranded DNA, ssDNA, or dsDNA, which is double-stranded DNA, whether RNA is single-stranded ssRNA or it's double-stranded RNA, like so. That's not in your syllabus. That was a little bit of unwanted excess information, but it's a very important classification system according to how viruses are classified. That's why I thought of mentioning it. And all you can see here is we have different proteins all around the virus. That's basically about viruses. And to finish things off, we will be doing a small comparison. Not a small, but an extended one, I guess, of eukaryotic cells and prokaryotic cells. Let me put that in the middle. Okay. We'll be talking about a certain feature, and then we'll see how prokaryotes fit into that feature, and then we'll see how eukaryotes fit into that feature. I think I'll draw this like that, and that all the way up until here. Let's join that up. Okay, cool. First of all, a feature. how it's affected in prokaryotes, how it's affected in eukaryotes, not affected, how it's different in eukaryotes, prokaryotes. Okay. First feature I'll talk about is the size. I don't want to really call it the size, therefore I'll call it the diameter. Prokaryotes are smaller than eukaryotes. That's the idea you have to get. They can range all the way from zero 0.5 to 5 micrometers. Eukaryotes can go all the way from 10 to 100 micrometers. That's the idea. What about the deoxyribonucleic acid or genetic material? Like we said, prokaryotes have what we call naked DNA. Naked DNA is simply found free in the cytoplasm and things are circular. For us, eukaryotic cells, what we have is linear DNA. We call what we have linear DNA. And it's surrounded by the nucleus. Our DNA is actually wrapped around these proteins. We call them histone proteins. More about histone proteins under cellular structures. All you have to know is there's something like this. There's a protein, that's our DNA. We take that and we wrap that around the histone protein. The ultimate goal of all of those things is to package the DNA into this tiny nucleus, which is about 6 micrometers in size. It's the only requirement of histone proteins. You will have to learn about that in a lot more detail than you think as you progress through your biology careers. So just remember that I told you something like that. Specifically about ribosomes. Simple important distinction. Prokaryotes have 70S ribosomes. Eukaryotes have 80S ribosomes. And there's a small size difference, okay? 70S ribosomes are a little bit smaller than the 80S ones. Okay, just a tiny tad bit smaller than the 80S ones. Let me extend these lines. Extend. Went too far, never mind. Let's keep it like that. Next, ribosomes. Endoplasmic reticulum. What do we have to talk about the endoplasmic reticulum? Whenever I drew a prokaryotic cell, did you find an ER? I do not think so. Prokaryotic cells are not complex enough to have endoplasmic reticuli. Of course, in eukaryotic cells, they are present. And you can either have ribosomes attached or not attached. If it's not attached, SCR, smooth endoplasmic reticulum. If they are attached, what you have is a rough endoplasmic reticulum. We call it the RER. A noteworthy mention about organelles. They are non-membrane bound. And here we have single organelles and they are double and non-membrane bound. Non-membrane. Okay, organelles are non-membrane bound or they are single, double or non-membrane. Okay, that's too confusing. All I'm trying to say is when it comes to prokaryotes, they only have non-membrane bound cellular organelles. They cannot have organelles that are membrane bound. But when it comes to eukaryotes, they have single membrane bound ones, double membrane bound ones, or no membrane bound ones. I should probably highlight it. They can have single membrane bound organelles, they can have double membrane bound organelles, or they can have no membrane bound organelles. Not a problem at all. Not a problem at all. Organelles, and let's talk about cell walls a bit. Final 2 points. When it comes to cell walls, basically, since we just talked about bacteria, we can say that cell walls are made out of peptidoglycan or murine. What about eukaryotic cell walls? Now, we talked about plants. No, they are made out of cellulose. Another eukaryote we didn't really talk about is fungi. Fungi cell walls are made out of this thing known as chitin. And since we're talking about cell walls and how things go, I'm next to that too. Okay, cool. The final mention under this would be cell division. How these things undergo cell division. Bacteria are very simple prokaryotic organisms. They can just basically split into two. That process is known as binary fission. While for eukaryotic cells, that cannot just happen. You can't just split the cell in two. You have to replicate organelles, you have to replicate DNA, and then only the cell can split in two. So we need complex processes like mitosis, meiosis, and cytokinesis. The cytokinesis is just the splitting of the cytoplasm, and I realized that nobody's going to understand that term if I write it like that. Cytokinesis. Key final takeaway: these are the comparisons between the prokaryotic and eukaryotic nature of cells. With that, chapter 1 is officially concluded. I'll just write over and chapter 1.